Hello, everybody. Today we will start lecturing on the replication of nuclear replicating large DNA viruses. These material can be found under lecture set number 12, large DNA virus. So we'll start by talking about herpes virus. Moving to slide number 2, the family Herpesviridae. Herpes is creeping spread of rash and vesicle lesions. Herpesviridae, a family of viruses, can be widely found in nature. They can infect plants, fungi, animals, and humans. Therefore, they have a wide host range. Herpesviridae virus family of viruses are highly infectious. These highly infectious viruses have different patterns of infection. The patterns of infection of Herpesviridae family of viruses include acute, persistent, and transform. There are 8 human herpes viruses, or HHV for human herpes virus, serotype 1 through H. Also, we have primate, bovine, equine, swine, marine, and avian herpes viruses. Moving to slide number 3, herpes simplex virus, or HSV1, infects mucous membranes and skin. It mainly causes infections in the oral and facial area, but the virus remains latent in neurons. Moving to slide number 4. Whereas herpes simplex virus 2, or HSV2, mainly causes infections in the genital area. Most of these infections are asymptomatic, meaning that individuals do not have clinical signs and symptoms of the disease, and at this stage they can be carriers. In instances that we see clinical signs and symptoms, the symptoms include genital lesions that will appear soon after exposure and last about 10 days. Herpes simplex virus remains latent in neurons, and most individuals, infected individuals, have recurrent episodes of HSV infection, especially within the first year of infection. Those mothers who have an active HSV infection may transmit the virus and the disease to newborns during delivery. And HSV infection is considered as an epidemic in the United States. It's estimated that over 20 million infections per year happen. Moving to slide number 5. The next virus is called varicella zoster virus, VZV. It is one virus that causes 2 diseases. These 2 diseases are called, these 2 viruses are varicella and zoster. Varicella is the causative agent or of chickenpox, or it causes chickenpox. The virus, varicella virus, remains latent in neurons, whereas the zoster virus causes shingles, and it's usually uncommon, but we see reactivation of the virus along nerve trunk in adults. So basically, you see a series of blasters along the path of a nerve in adults. Very, very painful because it damages the nerve, so it's very painful. Moving to slide number 6. Another virus within the herpes viridae family is called EBV, or Epstein-Barr virus. Epstein-Barr virus causes infectious mononucleosis, or IM. Epstein-Barr virus infects B lymphocytes, those lymphocytes that are part of the humoral immune response, epithelial cells, and fibroblast cells. So when we have the virus and an infection causes, since it hampers humoral immune response, the T cell immune response or cellular immune response kicks in. T lymphocytes recognize the virus, and virally infected cell results in the proliferation of the lymphocytes. And the virus, because it infects B lymphocytes, can remain latent in lymphoid tissues. Lymphoid tissues like lymph nodes, like spleen, like Peyer's patches in the stomach, are where the immune cells reside, and they first encounter antigen there. So the virally infected cells that are in the lymphoid tissue, so virus remains there, and later on in life, these patients, due to reactivation of virus or the presence of core carcinogen, cause BL, or Burkitt's lymphoma, a type of cancer of the lymphocytes, mainly B lymphocytes, or it causes nasopharyngeal carcinoma. Remember, I mentioned that the virus infects B lymphocytes, so those B cells can be cancers, we call them Burkitt's lymphoma, and epithelial cells of the nasopharynx can become cancers, we call it nasopharyngeal carcinoma. Moving to slide number 7. Another virus in this family is called HCMV, which is an abbreviation for human cytomegalovirus. Megalovirus, cyto, cells, megalo, large, so it's giant cells in culture. This giant cells or syncytia forms multinucleated cells, so multiple cells that each has their own nucleus will fuse together to form syncytia. Human cytomegalovirus is also a causative agent of infectious mononucleosis. It can infect monocytes, lymphocytes, epithelial cells. So you can easily imagine that a patient who is infected with HCMV is immunocompromised. Virus remains latent in the lymphoreticular cells. And in the United States, cytomegalovirus is a leading viral infection of fetus and newborns. Moving to slide number 8. Another virus is HHV6, or human herpes virus 6. It causes exanthema sebaceum, or roseola, which is a common rash in young children. and it infects lymphocytes. Slide number 9 is a little bit of a description of roseola or exanthema. I would encourage you guys to read through that description. Slide number 10, HHV7 or human herpes simplex virus 7. Human herpes virus 7 was isolated from lymphocytes of individuals who were contracted HIV, human immunodeficiency virus. HHV7 is an orphan virus. Orphan virus is a virus that is not associated with a particular disease, but still has the potential to be pathogenic. So it has pathogenicity. Moving to slide number 11, human herpes virus 8 infects lymphocytes, vascular endothelial cells, those endothelial cells that line the blood vessel. The DNA of this virus was found in a cancer that we see in AIDS patients called Kaposi sarcoma. Therefore, HHV8 is a co-carcinogen for Kaposi sarcoma, meaning that those individuals who have an infection with HHV8 are more prone to develop Kaposi sarcoma. So what is Kaposi sarcoma? A description of Kaposi sarcoma can be found on slide number 12. Moving to slide number 13, HSV1, human HSV1, human virus number 1. Sorry, guys. It has an envelope that this envelope has surface projections the size of human herpes simplex virus 1, human herpes simplex virus 1 is about 200 nanometer. It has a structure called tegument or matrix structure that is a structure between the capsid and envelope. This is a new concept that you have not been introduced to before. So we have capsid on the outside, and we have the genetic material inside. Between the capsid and envelope of the virus and the nuclear capsid, we have something called tegument structure. The nucleocapsid is icosahedral, and the core that contains viral DNA is wound in a cylinder shape. Slide number 14 gives you a description of what is a viral tegument or viral matrix. It's a very good tool, advantageous tool for the virus to not only be assisted in DNA replication, but also in evasion from the host immune responses. Let's move to slide number 15. Herpes simplex virus genome is a double-stranded DNA genome, is linear. One of these two strands has nicks or cuts in it. It's 150 kilobase, relatively large. The genome has two unique components called long and short, unique long or UL, and unique short or US, as we can see in the diagram above. It also has TR or terminal repeats on both terminal repeats S and terminal repeat R. Again, terminal repeat long and short, as well as IR or internal repeat sequences. And it also has a very conserved A sequence at both ends of the genome. This A sequence is used for recognition of the genome and insertion of the genome into capsid. So let's take another look at it. HSV genome double-stranded DNA virus, linear. One of the strands has cuts or nicks in it. There are some unique features to this double-stranded DNA genome. Number one, it has two unique components, two unique regions. One is larger than the other one, and one is smaller, so we call them unique long, UL, and unique short, US. We have terminal repeats. Again, one is long, one is short. Terminal, as the name implies, it's at the end of the genome. One is larger, the other one is smaller. And then we also have internal repeat sequences. As the name implies, they are not at the end of the genome. They are more internal within the genome. And we call them, again, they're long and short. We call them internal repeat, or IRL. And IRS also has a conserved sequence called A sequence, letter A, at both ends. This A sequence is needed for genome recognition and insertion of the genome into newly synthesized capsid. Slide number 16 talks about long terminal repeats. I am sure that you guys have been introduced to this concept in the prerequisite courses, but for those of you who, like myself, may have forgotten about this, here is a little bit of a reminder. Slide number 17 talks about HIV-1 because it has long terminal repeats. I really recommend you guys, you have time, so read through this because in a couple of lectures we'll talk about HIV, and it's a little, the replication is a little bit of interesting, to say the least. So I want you to be familiar with that. Moving to slide number 18, it is herpes simplex virus genetic and transcription map. So it basically shows the relative location of each transcriptional unit and then the factors that contribute to the expression of various genes. Moving to slide number 19, this is a very, very, very important slide. Again, very important slide. How HSV gains entry into the cell and uncoats, meaning that its genome is being separated from its capsid. Capsid gets into the cytoplasm. Once it gets into the cytoplasm, remember we said that there is a matrix structure or tegument. This tegument is called VHS, virion host shutoff. So what does it do when capsid gets into the cytoplasm? Capsid and genetic material, capsid gets in, releases the tegument VHS, and tegument degrades host cell messenger RNA. So then host cell proteins cannot be translated. Then this viral capsid will be transported to the nuclear membrane, and Once it is at the nuclear membrane, it kind of releases viral DNA along with another factor called alpha TIF into the host cell nucleus. Alpha TIF stands for alpha trans-inducing factor, or we call it VP16, is a trans-acting protein. What does it do? It comes from the virus. It forms a complex with host transcription factors. Host transcription factors are called OCT1 and HCF. A complex composed of alpha TIF coming from the virus, and two host transcription factors, OCT1 and HCF, induce immediate early gene transcription in the herpes simplex viruses. So let's take a step back. Virus gets into the cytoplasm. Once the viral capsid is in the cytoplasm, it releases a tegument protein called VHS that is called a virion host shutoff. This tegument, VHS, what it does, it basically degrades host cell messenger RNA, so host cell proteins cannot be translated. Then viral capsid will be transported to nuclear membrane. At the membrane that surrounds the nucleus of host cell, the viral DNA and another factor called alpha TIF, or trans-inducing factor, will be released into host cell nucleus. Alpha TIF from virus will form a complex, will associate with OCT1 and HCF transcription factors. These are protein-protein interactions. This complex, composed of alpha TIF, OCT1, and HCF, results in activate immediate early gene transcription in HSV. Moving to slide number 20, I have a detailed description of entry of human herpes simplex virus into a host cell. So please take a look at this slide and go through some of these details. Slide number 21, once the virus gets in, how the messenger RNA of HSV will be transcribed. DNA genome circularizes. So the promoter and enhancer sites will become activated by that protein complex made up of viral alpha TIF and some host cell DNA binding proteins such as OCT1, SP1. And we will have transcription from both DNA strands. Remember, I said that HSV genome is double-stranded DNA. One strand has NICs. So transcription would be bidirectional, both directions. One goes clockwise, the other one goes counterclockwise, because the genome will circularize. And viral mRNA transcription requires host cell RNA polymerase II. Moving to slide number 22. So HSV has a very regulated pattern of gene expression, meaning that at different time points, different genes are being expressed. These can be divided into three phases: immediate, early, and late. Immediate early gene expression results in production of alpha gene products that are mainly regulatory, whereas early gene expression results in beta gene products that these are mainly viral enzymes and proteins that are required for DNA replication. And late gene products are gamma gene products. Some gene products are regulatory proteins, but mainly structural proteins. Again, during early, we provide those enzymes required for DNA replication, and during late gene expression, we make capsid proteins so genome and capsid can associate to form new virions. Moving to slide number 23, this is basically a cascade of HSV transcription. The letter U's that you see at the bottom are the units, transcriptional units. Immediate and early gene products usually use those gene segments closer to IRL, IRS, unique short, and terminal repeat short, whereas early uses these transcriptional units as well as those located in the other regions of the genome, and late also uses the same but at a more robust and enhanced level. Moving to slide number 24, very important slide. DNA replication uses a rolling circle method. So the synthesis of DNA in a long strand, it's long strand, so it's bits and pieces. Head of one piece is attached to the tail of another piece, so we call them head to tails, and they form concatenars. Remember, I told you guys earlier on that one of the strands has nick. The nick DNA strand is a single-stranded DNA at the 5' ends starts rolling off. So we have two types of DNA synthesis: continuous, which is the outer strand that copies the inner circular strand and extends from the 3' end. And also we have discontinuous DNA synthesis that occurs on that DNA strand that has a nick on it and has rolled off. We make a new complete new strand that the combination of discontinuous and continuous forms a double-stranded DNA. These DNA, newly synthesized DNAs, are in the shape of concatenars that they are later on cleaved into genome-size segments, and this process is aided by recognition of that A sequence or A termini that we discussed before. Slide number 25 describes rolling circle replication for those of us who may have forgotten how it is from other courses, which slide number 26 talks about what is a concatemer and how they play a role in human herpes virus 6 replication, etc. I want you guys to take a look at this and refresh your memory from the previous prerequisite courses. Moving to slide number 27, again, a very important slide. I had mentioned before that HSV can cause latent infection. How does it cause latent infection? Virus ascends up the sensory nerve and it goes to neuron. The viral DNA is associates or complexes with host cell histones and will set up shop or will establish in host cells as episome and extra chromosomal segment. Virus expresses LAT or LAT, which stands for latency associated transcripts. These are those transcriptions that are these are those transcripts or mRNAs that are required during the latency period of the virus. We do not see any infectious virus replication going on. We just see viral DNA. And so virus remains latent. Then it can become reactivated. How virus gets reactivated? When there is stress stimuli, there is an injury, UV light exposure, hormonal imbalances, and most importantly, immune suppression. All of these result in the reactivation of a virus. So virus remains latent, then it becomes active again. Moving to slide number 28, there are a set of 6 very useful YouTube links. As I've told you before, I've gone through a large number of YouTube video clips and handpicked a handful of them that would shed more light on the material that we discussed during the lecture. I strongly encourage you guys to take a look at these video clips. Slide number 29 is a summary of the herpes virus lecture. We have 8, herpes virus has 8 human members. These are HSV 1 and 2, herpes simplex 1, herpes simplex 2, varicella zoster virus, one virus that causes 2 infections, varicella chickenpox and zoster or shingles. EBV Epstein-Barr virus, HCMV human cytomegalovirus, HHV or human herpes virus 6, 7, 8. HSV uses host cell RNA polymerase II to transcribe its messenger RNAs. The messenger RNAs are immediate early within minutes. They produce alpha gene products, early produce beta gene products, and late early gene products beta are mainly enzymes required for DNA synthesis, and late gamma gene products that are mainly structural proteins. A rolling circle model for replication of HSV DNA into long concatemer is being used, that the long concatemer will later on be cut into separate genomes, so smaller fractions. Late infection of herpes virus in neural or lymphoid tissue can occur. Virus remains latent, ascends then neurons, and this is due to the expression of a protein called LAT, or latency associated gene expression. The reading material associated with this lecture can be found in chapter 17. And I will stop here. The material we covered in this lecture, large DNA viruses, are not part of your second midterm. Thank you so much for your attention. Please go over the lecture materials a couple of times, and if you have any questions, then feel free to contact me. Have a wonderful day and stay safe and healthy out there. Take care, and I'll hope to see you guys soon.